Hello and welcome. Today we will explore the different approaches scientists use to study microbes. Why is this important? Because microbes are the most abundant and diverse life forms on earth. They exist everywhere in soil, water, our guts, extreme environments like hot springs, as well as from human body to deep sea vents to deserts and glaciers. But how do we study something so small, so diverse, and so often invisible? Studying microbes is challenging because most of them are invisible to naked eye, and many cannot be easily grown in laboratory. Our ability to study them depends heavily on the tools and approaches we use. Over time, scientists and microbiologists have developed two major approaches to studying microbes. One is culture-dependent methods, where microbes are isolated and grown in laboratory conditions. Another is culture-independent methods, where microbes are studied without culturing, usually using molecular or sequencing-based techniques. Historically, microbiologists relied on growing microbes in the lab. This is called culture-dependent approach or method, but with the advances in molecular biology and sequencing, we now have culture-independent methods that don't require growing microbes at all. Nowadays, culture-independent or molecular or sequencing-based approaches are dominating the microbiology research. In this lecture, we will explore these approaches, understand their strengths and weaknesses, and see how they complement each other in modern microbiome research to study microbial communities comprehensively. This lecture is important because it shows us how microbiology evolved from petri dishes to sequencing machines. Now we are going to discuss culture-dependent methods, one of the earliest and most fundamental approaches to studying microorganisms. Although modern microbiology often uses culture-independent methods like sequencing, culturing remains the gold standard for functional studies, antibiotic testing, and industrial application. Now let's look at the key steps involved in culture-dependent methods. The first step is sample collection. Microorganisms are collected from different environments such as soil, water, air, food, or clinical specimens. These samples may come from natural ecosystems, industrial settings, or from human body. The goal is to obtain microorganisms from their original environment so they can be studied in laboratory. The second step is inoculation on culture media. Inoculation simply means introducing microorganisms from the collected sample into a nutrient-rich culture medium that supports microbial growth. The sample is transferred onto or into the culture medium under controlled laboratory conditions, allowing microorganisms to grow and form visible colonies. Culture-dependent methods rely heavily on different types of nutrient media, each designed for specific purposes. One common type is general-purpose media. General-purpose media are nutrient-rich culture media designed to support the growth of a wide variety of non-fastidious microorganisms. These media contain essential nutrients such as carbon sources, nitrogen, minerals, and vitamins. Examples include nutrient agar and LB broth, which is commonly used for microbial culture. Another important category is selective media. Selective media are specialized growth media that contain specific substances such as dyes, antibiotics, salts, or particular nutrients that inhibit the growth of unwanted microorganisms while allowing or permitting the growth of specific or targeted microbial groups. This is especially useful when scientists want to isolate a particular organism from complex microbial community. For example, McConkey agar selectively supports the growth of gram-negative bacteria, while mannitol salt agar favors the growth of staphylococci due to its high salt concentration. Next, we have differential media. Differential media are designed to distinguish between different microorganisms based on the biochemical characteristics. This difference may involve processes such as fermentation, enzyme activity, or hemolysis. The results are often visible as color changes, variation in colony appearance, or pH shift in the medium. Unlike selective media, which inhibit or promote the growth of specific bacteria, differential media usually allow many organisms to grow but provide visual indicators that highlight their differences. For example, blood agar is widely used to observe different types of hemolysis, which can help identify bacterial species. Another important type is enrichment media. Enrichment media are typically liquid culture media designed to enhance the growth of a specific microorganism or group of microorganisms. They provide favorable nutrients and environmental conditions that allow target organism to grow more rapidly, increasing its relative abundance in a mixed sample. Unlike selective media, which directly inhibit unwanted microbes on solid agar, enrichment media usually work in liquid form, giving the target microbes a competitive growth advantage without necessarily killing others. This approach is particularly useful when the target microbes is rare or slow growing. For example, selenite F broth is commonly used for enrichment of Salmonella species from fecal sample. In this medium, sodium selenite inhibits coliform bacteria and other competing gut microbes, giving Salmonella a growth advantage. After inoculation, the next step is incubation under suitable environmental conditions. The inoculated culture media are placed in incubator where temperature, oxygen levels or other environmental factors are carefully controlled. These conditions are adjusted depending on the types of microorganism being studied. For example, human pathogenic bacteria are commonly incubated at around 37 degrees Celsius, which corresponds to normal human body temperature. In contrast, many environmental microorganisms grow better at slightly lower temperatures, typically between 25 and 30 degrees Celsius. Some microorganisms require special atmospheric conditions, such as anaerobic bacteria which can't tolerate oxygen must be grown in anaerobic chamber or jars where oxygen is removed. On the other hand, aerobic bacteria may be grown in shaking incubator which continuously mix the culture and supply oxygen. Once incubation is complete, the next step is observation of the colony morphology. After the sufficient growth, the culture plates are examined for colony growth. When microorganisms multiply on solid culture media, they form visible clusters of cells called colonies. These colonies often show distinctive characteristics. They may differ in size, shape, color, margin, elevation, or texture. Observing these features provides preliminary clues about the identity of the microorganism. Importantly, each colony usually originates from a single microbial cell, meaning that the colony represents a clonal population of genetically identical cells. For example, Staphylococcus aureus typically produces golden yellow colonies, while E. coli forms off-white or cream-colored colonies. Although colony morphology alone cannot provide complete identification, it offers important initial information about the microorganism. The next step is isolation of pure cultures. It refers to the process of separating a single microbial species from a mixed population so that only one type of microorganism is present. In most natural samples, microorganisms exist as mixed population. Therefore, microbiologists must isolate individual species in order to study them accurately. A pure culture contains only one type of microorganism. All the cells in a pure culture are genetically identical and derived from a single parent cell, forming a clonal population. To obtain pure culture, microbiologists use several standard laboratory techniques. One of the most common techniques is stacked plate method. In this method, a sterile inoculating loop is used to progressively spread and dilute microbial cells across the surface of an agar plate. As the cells become more diluted across the plate, individual cells eventually grow into separate colonies. Another technique is the pour plate method. In this method, the microbial sample is first serially diluted, then mixed with molten agar and poured into petri dishes. As the agar solidifies, microorganisms become trapped within the medium and grow into colonies throughout the agar. The third technique is spread plate method. In this method, a small volume of diluted microbial suspension is placed onto the surface of an agar plate and evenly spread using a sterile spreader. The microorganisms then grow as separate colonies on the surface of the agar. These techniques allows microbiologists to isolate individual colonies and obtain pure cultures, which can be used for further identification and analysis. After obtaining pure cultures, the next step is identification of microorganisms. At this stage, microbiologists determine what organism has been isolated. Identification usually involves a combination of morphological observation, biochemical testing, and molecular techniques. Let's begin with identification. The approach is based on microscopic and macroscopic characteristics of microorganisms. Using a microscope, scientists observe the shape, size, and arrangement of microbial cells. Bacteria may appear as spherical, rod-shaped or spiral-shaped. They may also form specific arrangements such as strains, pairs or clusters. Staining techniques are also very important in microbial identification, which differentiates bacteria into gram-positive and gram-negative group based on their cell wall structure. For example, Staphylococcus aureus typically appear as gram-positive Koch arrangement in cluster, whereas E. coli appears as gram-positive rod-shaped bacteria. Another example is acid-fast stain, which is commonly used to identify bacteria such as Microbacterium species. In addition to microscopic features, microbiologists also examine colony morphology on agar plates. Colonies may differ in size, color, age, elevation, texture, providing useful preliminary clues about the microorganism. However, morphological features alone are usually not enough for complete identification. Therefore, the next step often involves biochemical characterization. Common biochemical tests include catalase and oxidase tests as well as sugar fermentation tests. Other tests may detect urease activity, indole production or hydrogen sulfide production. In clinical laboratories, identification can also be performed using automated biochemical systems. These include systems such as API strips or BITEC. Another modern approach involves advanced automated technologies. One powerful method is multi-top mass spectrometry, which identifies microorganisms based on their unique protein fingerprint. This technology allows rapid and highly accurate microbial identification and is widely used in many clinical microbiology laboratories. Finally, one of the most accurate methods is molecular identification. Molecular techniques analyze the genetic materials of microorganisms. One widely used method is polymerase chain reaction or PCR, which amplifies specific regions of microbial DNA. Another highly reliable technique is 16S ribosomal rRNA gene sequencing, which is considered the gold standard for bacterial identification because the 16S rRNA gene is highly conserved among bacteria, but it also contains variable regions. Sequencing these genes allows precise identification of the bacterial species. In many modern laboratories, once a pure culture is obtained, 16S rRNA sequencing is often used to confirm the identity of the microorganism or bacteria. The culture-dependent methods have several important advantages. First, they provide live microorganisms, which allows the researchers to study their growth, metabolism, and physiology. Second, these methods allow testing of antibiotic resistance and pathogenicity. They also enable genetic and functional experiment. Importantly, the culture methods allow pure culture isolation, which is essential for cost postulates. Finally, cultured microbes are widely used in industrial and medical applications, including antibiotic production, fermentation, and probiotic development. Although the culture-dependent methods have been the foundation of microbiology for more than a century, they also have several important limitations. One of the most well-known limitations is great plate count anomaly or low culturability. When scientists collect an environmental sample such as soil, seawater or even samples from the human gut and examine it under a microscope, they can observe thousands or even millions of microbial cells. However, when the same sample is placed on agar plates in the laboratory, only a very small fraction of these microbes actually form visible colonies. For example, imagine taking a single drop of seawater. Under the microscope, you might see thousands of microbial cells, but when the same drop is cultured on a plate, only a few colonies appear. This means the vast majority of the microorganisms present in the sample remain undetected using culture-based methods. In many environments, only about 1 to 10% of microorganisms can be cultured using standard laboratory techniques. This huge gap between the number of cells observed microscopically and the number that grow on plates is what we call the great plate count anomaly. For example, for decades, scientists believed the human gut microbiome contained only a small number of dominant microbial species because those were the organisms that could be cultured in the laboratory. However, with the development of DNA sequencing technologies, researchers later discovered thousands of different microbial species living in the human gut. Another reason for the limitation is that many microorganisms have very specific growth requirements. They may depend on particular or unknown nutrient, precise environmental conditions such as temperature, pH or even interactions with other microorganisms or symbiotic partners. These complex natural conditions are often difficult to reproduce in the laboratory. In addition, culture-dependent methods often show culture bias. In mixed microbial communities, fast-growing microbes tend to dominate the culture plates, while the slow-growing organisms may be outcompeted and remain undetected. For example, the pathogen Mycobacterium lepra grows extremely slowly and may take weeks to months to be detected. Because of these challenges, culture-dependent methods often fail to represent the true diversity of microbial communities in the natural environment. Another limitation is that culturing microbes can be time-consuming and labor-intensive, as isolation, purification, and identification may take several days to even weeks. Another challenge is that many microbes exist in dormant or viable but non-culturable state, meaning they are alive but cannot grow under standard laboratory culture conditions. In the next video, we will explore culture-independent methods or approaches in more detail. So here we have discussed the approaches to studying microbes focusing on culture-dependent methods. We covered the key steps from sample collection to isolation, growth, and functional studies. We also highlighted the main advantages such as working with live microbes, performing physiological and genetic studies, and supporting industrial application. Finally, we discussed the major limitations including low culturability, specific growth requirements, slow growing microbes, and the fact that these methods cannot capture the full diversity of microbial communities. Overall, we can say while culture-dependent methods remain foundation in microbiology, they are often complemented by modern culture-independent approaches to provide a more complete understanding of the microbial world. Thank you everyone.